Okay. Yeah, so see, we are going live. We are live on YouTube. I will move to, can you see the starting slide? Okay, so I'm beginning the webinar. People will be coming in now. The robot is saying this meeting is being recorded. So. So we'll wait a couple of minutes for the people to come in. So we'll wait a couple of minutes for the people to come in. Debarshini, could you mute yourself? Thank you. Okay, so I will just begin the housekeeping instructions. So welcome everyone to our CTIS Tech Platform Series webinar. So these are basic housekeeping instructions for all of you who are watching on Zoom. If you're watching on YouTube, just we will get to you. First thing is the audio settings. Later on, we may want you to speak. So make sure that your mic is working. So in the webinar, if you can, please go to the bottom left-hand corner, and click on the button called audio settings. This is a screen grab of how Zoom looks like on the browser or your app. And once you're there, you can check your speaker and microphone, etc., and make sure it's working. So then when we raise your hand, the call will go smoothly. The next bit is the raise hand icon. It's shaped like a hand, and you click on it, it turns green, and your name goes to the top of the list. So this enables us to find you very quickly in case of this webinar, there are many participants. So you need to raise your hand to make sure your name is at the top of the list so we can unmute you. Also, most importantly, the Q&A box, it's got a Q on it and it's labeled Q&A. You can click on the Q&A button and you can put in your question. Your name will also be there in case you are afraid of your name being known. You can click the send anonymously box, the send anonymously option, and click send, and we will never know that you have asked the question. To date, we have not figured it out yet. Not that we have tried. After that, Q&A for anyone listening on YouTube. We will just request them to put your questions there in the comment section. If you're listening on CCamp or on the BLiSC India YouTube channel, please just put your questions in the comment. We will copy paste them and we will get to them as many as possible. So that is about it. I will now hand it over to Dr. Debarshini for formally introducing the session. Thank you so much. And if you have any issues or you can't hear something, just please put it in the Q&A box. Chat has been disabled.
Thanks, Chandrakant, for hosting today's webinar. Very good morning to you all, and welcome to this 4th edition of the CTIS Tech Platform Series. Most of you who are here are already aware that CCAMP, or Center for Cellular and Molecular Platforms, is one of India's most exciting bioinnovation hubs, where we promote cutting-edge research and innovation in the life sciences. It is a part of the Bangalore Life Sciences Cluster, and the cluster, the reason I mention it is here, which will become clearer as we go on, is that the cluster is a campus that houses state-of-the-art technology platforms for access by scientists and researchers across India. And today we are here for a webinar on genome engineering through mouse and Drosophila research models. The webinar is a part of the CCAMP Technology and Innovations Webinar Series, or CTIS, which is an attempt to facilitate interaction between you, the audience, and the people who are manning our various programs and facilities. And CTIS, or the CTIS Tech Platform Series, is a focus series under the same umbrella. And in this series, what we are doing is we are bringing a set of talks on these various cutting-edge technology platforms that I said just now are housed at CCAMP and BLiSC, and what are their myriad research applications and what kind of services can you access with us. And of these technology platforms, we have state-of-the-art transgenic animals facilities at BLiSC, consisting of 2 primary facilities. One is the Mouse Genome Engineering Facility, or MGEF, and the Transgenic Fly, or Drosophila facility, which is the only publicly available Drosophila facility in India currently. And from genetically modified mouse models to cryopreservation to genome editing technologies such as CRISPR-Cas9, these 2 facilities have it all. And in today's session, BLiSC experts will tell you more about these platforms, their applications, and how you can access these services through CCAMP and BLiSC. And with that, I now pass on the mic to my colleagues, Dr. Aurelie Jory, head of the Mouse Genome Engineering Facility, MGEF, at BLiSC, Dr. Deepti Trivedi, head of the Transgenic Fly facility at BLiSC. But first, I will hand over the mic to Dr. Atif, program lead of technology platforms at CCAMP, to give you an introduction to what we do and who we are at CCAMP. And over to you, Atif.
Thank you, Debarshini. Good morning, all. Thanks for hosting the seminar, and welcome all on this lovely morning in Bangalore. I'm sure wherever you are, you're all safe and sound. So I'll quickly give a brief overview of what we do through CCAMP and Bangalore Life Science Cluster. I'll keep it very short so that we come to the topic at hand today, which is the genome engineering using the most prominent animal models, that is mouse and Drosophila. So quickly, I'll get going to give you a glimpse of overall what we do at CCAMP and the Bangalore Life Science Cluster. CCAMP was started by the Department of Biotechnology to be an enabler of cutting-edge research and innovation in India. And it is housed within this really exciting life science cluster called the Bangalore Life Science Cluster, having 3 institutions: National Center for Biological Sciences, which is a premier life science research center focused on cutting edge fundamental research. And then the other constituent is the Institute for Stem Cell Research and Regenerative Medicine, INSTEM. It's India's only federally funded institute to do stem cell and applicable research in regenerative medicine. And CCAMP, as I mentioned, is an enabler organization where we do a couple of things in 3 large verticals. We provide technology resources such as mouse and Drosophila genome services to all researchers across scales. Then we do our own in-house R&D, that is translational research for taking academic discoveries to market-ready technologies. And then we have an entrepreneurial program where we support life science innovation, entrepreneurs, and startups. Just to give a glimpse of what we do in entrepreneurship is we start with identifying exciting ideas, we fund them, we incubate them, and then we accelerate them. And in a way, if you see the whole journey of a startup, right from finding the exciting ideas, supporting them at a very early stage with grant funding, seed funding, connecting them to mentors and networking, avenues, and giving them the aspects which are perhaps not very natural to life science researchers, that is the regulatory, the funding, the investment and other opportunities, bringing them larger tickets of fundings and accelerating them through various accelerator programs. So there's a glimpse of the various funding opportunities available through SRICAM, which are both a mix of government fundings as well as private fundings. And the fundings are also available at various scales of requirement, from all the way early stage funding, bridge funding, to accelerator, large scale fundings. I'm really excited to share that some of our startups have been very successful in raising Series A fundings, which actually run into a couple of $1 million. So that's the kind of span we can support life science innovators and startups through CCAMP. Of course, it goes without saying that all this requires a laboratory space, which we also offer through our state-of-art bioincubation facility at CCAMP. And then there are a variety of networking opportunities across the spectrum. Ideally, we would love to have go back to this lovely time where we would meet up on open spaces and meeting rooms and put you in touch with one-on-one with the the subject area experts and investors. These days, in the new normal, we do it online. Hopefully, things going well, we'll start seeing this back again. So here I'll stop and shift gears to the technology platforms that we have in this campus. We are very lucky to have a huge portfolio of resources and technologies at our disposal, all largely funded thanks to the various funds that we received from time to time from the government agencies. And a very, very important aspect of it is that we make these technologies available to researchers in every domain, be it academics, be it startups, or established industry. And I'll give you a very speed tour of the facilities that we have on campus, beginning with next-gen genomics, where we have small-scale and large-scale genome sequencers, which can handle a variety of applications like whole genome sequencing, transcriptome, ChIP-seq, etc., to a very large portfolio of mass spectrometry services. And we have expertise in a variety of omics within the mass spectrometry domain, including proteomics, metabolomics, glycomics, lipidomics, as well as something which is very, very interesting and exciting for biopharma industry, that is a dedicated biologics characterization facility, which again offers a variety of services and end-to-end services for characterization of the large molecule biotherapeutics that are being developed. India is a very large developer of biotherapeutics and vaccines, and these are supported by this facility in terms of their development and the scale of characterization before regulatory filings. Then we also have the more facilities which support molecular and structural studies like NMR, X-ray crystallography facility, cryo-electron microscopy facility. This is perhaps, till the last I checked, the only 300 kV electron microscope in India, which can support structural determination up to Armstrong level. So you can have biomolecular structure in a highly resolved manner. And then the flow cytometry facility again has a number of flow cytometers, which again support large analysis types, both analysis as well as cell sorting. And we are, as I said, really fortunate to have one of the most advanced state-of-art imaging facilities at our disposal, which includes confocal imaging, fluorescence and super resolution microscopy, scanning electron microscopy, transmission electron microscopy, and 3D X-ray microtomography facility. And some of the examples are here where you could do really high resolution imaging as well as optical based correlation studies as on a variety of matrices, not just biological samples, but also samples like drug emulsions and nanoparticles. And this is a hair, as well as you could do a whole organism imaging by the 3D X-ray tomography. And a couple of core equipment that are a constant requirement in any biological lab that are also available and accessible to researchers. And in this time, we could not have not served the really burning need, and that is the assays against SARS-CoV. So we do have access to viruses and pseudoviruses, and we can do we can do COVID assays on a variety of starting materials like formulations, drug compounds, drug products, devices, etc. As we are growing in this field of biology, a lot of integration of machine learning, AI, medtech, and diagnostic is coming together. And for that, we are cognizant of it, and also, again, fortunate to have been funded by the Department of Biotechnology and BIRAC to set up this integrated facility for designing, developing, prototyping, and pilot scale manufacturing of microfluidic devices for diagnostic purposes. But today we are here to hear from the experts of mouse and Drosophila facilities, and we cannot emphasize more how important model organisms for understanding physiology and disease mechanisms and disease biology when you are either doing basic research or you are developing a drug product or any of the applications which eventually has a use in human biology understanding. Therefore, this is one of the key aspects, and we are again fortunate to be able to set up these facilities. And over the years, they have serviced a lot of internal as well as external researchers. And I'm excited to have the heads of these facilities, Dr. Aurelie and Dr. Deepti, to share the most exciting work that we are doing, particularly on the genome engineering aspect using largely CRISPR-Cas9 methods. Before I hand it over to my colleagues for giving you a detailed presentation and also take your questions more importantly, just a glimpse of what work has come out of these facilities. This is something which is an internal work using cryo-electron microscopy. One of the groups in Hyderabad has published their work that was partly supported by the Mouse Fly facility in this journal early this year. And another group from IIT Guwahati used the proteomics facility, imaging and flow cytometry facility to come up with an exciting work. These all publications are indicative of how important it is to use these technologies in order to answer some of the sophisticated questions we are asking, and how we in the campus are able to support these. All these publications that have come through and have used our facilities are listed here in this website. Feel free to go and look at it up and see what kind of work has already been done, and maybe it will give you ideas to plan some of your own experiments. Before I sign off, there is one aspect, and that is that all these things are good and great, but how are we making them beyond services, and offering them beyond services? And that's an aspect of training. And this is another very key mandates that we have on the campus to make these facilities not just available, but also train people on them. And we have these perennial training programs which happen and are listed on this website. Ideally, they are hands-on trainings, but given the current situation for the last year and a half, we have not been able to do anything in-house, in person. But then we have pivoted and started doing webinars, web-based trainings, and that will continue till such time as it is safe for everyone to have in-person trainings. So have an eye on this web page and our media handles to be able to be aware of what is the next upcoming program, and we would be delighted to have you on on one or many of these training programs. With this, I'll stop here and hand it over to my colleagues. But in case there is a quick question or two, I'll be happy to take. Yeah. If not,
I think, yeah, Atif, I think we can move on possibly.
Sure. Thanks. Oops. Yeah. Okay. Okay, so I'll be very happy to invite Dr. Aurelie Jory. She is the head of the Mouse Genome Engineering facility and co-head of the Animal Care and Research Center here at NCBS and Bangalore Life Science Cluster. A quick background about Lily. She received her master's and PhD degrees from Pasteur Institute, Paris, France. Then she had a stint at Pasteur Institute and also at Columbia University before coming to Bangalore in India and back in 2014 to set up this very exciting DPT funded National Mouse Resource Initiative. And since 2014, she has been here with us to basically set up and head this really exciting and perhaps again the only publicly available mouse facility in the country. Over to you, Lily, and a very warm welcome.
Okay, hello everybody. Hope you all well. So let me start by sharing the screen. So yes, like Atif mentioned, so I've been here since 2014, and the goal of me coming here was really to set up this mouse genome engineering facility. This was under a national, it is still under the national project funded by DBT. So let me try to put my full screen. Should be okay now, right? Yeah. Okay. So I'm gonna talk a little bit about the different types of services. So I know this seminar is mostly focused on the on the genome engineering part of our services, but I'll quickly go through the other couple of services that we provide here at Mouse Genome Engineering Facility. And the reason for that is that it's kind of inclusive. When you have a very nice and important mouse model, you need to make sure that it's safe, whether it's in a live status or in a cryo status. And you need to be able to maintain it properly over the years. So for that, here is a graph of our number of projects that we've catered, both, like Atif said, internally and externally. In our case, it's mostly external clients, as for that matter. And so here is the number of projects and no mouse models that we have generated over the years. Obviously, it takes much more time to generate these models, so there's not as many as other services that you can see here. So for example, let me try to, yeah. So the most common service that we provide is a sperm or embryo cryopreservation services. And this is actually very important, and it has turned out to be actually crucial during these lockdown times. So basically, each time we import new live mice stocks here at BLiSC, we have this habit to cryopreserve them by meaning cryopreserving their sperm or their embryo as fast as possible. So what is it, Deepti? It's okay, no?
Yeah, just go ahead. There is.
Okay. So that I'll explain a little bit how we do that. And then similarly, we should be able to cryorecover the stocks that we cryopreserve, and this is done by either IVF or regeneration services. And again, this has been crucial. Many people who have, their facilities had kind of slowed down or completely shut down in some cases due to the lockdown and the COVID status. They lost many of their precious stocks, or the stocks stopped breeding because they had not been breeding for a long time. And so we have been actively able to revive these stocks for people here on campus, but also a lot for people outside who had the great idea of having preserved their stocks ahead of time. Training, as Atif mentioned, we are very, we do a lot of training. We did a lot of hands-on training. Hopefully that will happen again soon, but we are now working on doing video-based training programs as well to be able to do this online. Okay, so first let me just go over a little bit the cryobanking services that we have here. So basically, if you have, and why it's important, it's not only to preserve your stocks during disaster, to be able to recover them in case of disaster. It's also to preserve the stocks from preserving their genetic quality. As you breed, whether it's an animal, a fly, or a cell, you will have spontaneous natural mutations that occur, and that can, over time accumulate in your organism and eventually, change the status of your model. And so to be able to come back and go back to the original cryopreserved genetic material is very important in these cases. So we cryopreserve sperm, and this is a mouse sperm. The mouse sperm has this very unusual crooked head. Looks kind of evil there, but it's actually very nice. And so we freeze them in these little, straws. I don't know if you can see. That goes into the liquid nitrogen. And then we're able to recover these sperm aliquots and do an IVF and reimplant the fertilized embryos into a pseudo-pregnant female and recover live animals from that. We keep all of this data, the stocks that we have here at BLiSC, either live or in cryopreserved forms in a national database that you can access from our website. And basically, if you're interested in one of these stocks, you can email us. Obviously, there might be some MTA regulations for some of them, but usually that goes pretty well. Embryo, same thing, so we vitrify. So it's slightly a different technique from sperm and embryo, the way we freeze them down, but we're able to freeze and revive them as well. So why sometimes sperm and sometimes embryos? That I can answer that later on if the question pops up, but it really depends on the genetic, the mutation that you want to preserve. If it's very important to have it at the homozygous stage, for example, then you need the embryo to be vitrified. If you just want to preserve the allele, then sperm is just fine. Sperm is much easier to preserve and cheaper than embryos. So when embryo are not necessarily required, by default, we do the sperm cryopreservation. So in vitro fertilization, like I said, that's a service that we do routinely here. Basically, we do it first to quality control that our sperm is properly cryopreserved and that we can revive it. So when the client comes to us and provides us a couple of males to freeze down the sperm, we have usually about 10 straws per male of sperm. And so we always take one aside and we thaw that sperm, do an IVF, and that's our quality control IVF. We make sure that the sperm is active, that it's able to fertilize the embryos, and that it goes up to the blastocyst stage. For some rare cases, we actually go to the implantation and live PEP. It depends on the complexity of the model and the mutation. And so this is how we do it. And we have a threshold. So the success of IVF really depends on genetic background. Some strains do better than others. We always run in parallel a control IVF with our wild type sperm. So that gives us a threshold of, make sure that the IVF session actually went well. In case the client's sperm IVF is of low QC, it's not because we did something wrong, it's just because of the genetics of the animal is a little bit lower. And that's fine. In that case, we know that at the time of revival, we just need to do a little bigger IVF session to have higher numbers of embryos to reimplant. So I'm not going to go through much, but basically we've done a lot, and depending on the background and the transgenics. So here for each pair, you have the control IVF on the left. So, some of for the wild type, it goes almost sometimes to 100%. In some transgenic models, it shifts a little bit lower. But overall, in very rare cases, are we below 30% in what we do, which means that we are able to revive any stock that we freeze down. This is the embryo cryorecovery, so we have less demand for that, but that works well. We also, so the embryo recovery that we do for now, most of the recoveries that we have done are for clients that import embryos from these international consortiums. So there's many consortiums around the planet that have stocks of embryos, of mouse stocks, sorry, and they're preserved as embryos. So they import the frozen embryos to our facility, and we're able to revive these lines from the cryopreserved embryos. So how do we do? So once all of this, what I've talked so far is kind of in a dish, in vitro situation, but at the end we want a live animal to be able to do our experiments. So once we have the embryos, whether they're from natural mating that we need to...
Lily, if I can interrupt, so sorry. What's happening is people are not seeing the edge of your slides. They are able to see it on YouTube, and they were seeing it for a while, but they're not seeing the edge of your slides. So if you could move out of presentation mode.
Sure.
Just for and they will be seeing the side bits of your...
Okay. How do I close this?
Yeah, I think this is better.
Yeah, and if you can just allow them to see it, not need, maybe zoom out,
Zoom in.
not zoom in.
Yeah.
Zoom out.
Zoom out again. Yeah, is this okay?
Zoom out. Yeah, this is good. And so this will allow them to see the whole slide.
Okay, sure.
Okay, thank you everyone. We apologize for the technical problem. This happens.
So the embryo transfer mechanism is to actually go from an in vitro cultured embryo, whether it's from the IVF session, the embryo cryorecovery, or the in vitro maintained embryo, pre-implantation embryo. So this means that it's only from one cell to blastocyst. And then we load these embryos into a little capillary with some media, and we take a female that's been stimulated for pregnancy by vasectomized male. That's a little technical. And we do a live surgery. So we anesthetize the female, and we kind of re-implant the embryos in the ampulla of the oviduct of the female. We close everything down. The animal wakes up and carries out the pregnancy with the modified or the IVF generated embryos, and this works out pretty well. So we've been doing a lot of that. Okay, so now the genetic engineering part. So we have set up this facility, and exactly when I arrived, and I think it's kind of still the case, there are many institutes a few institutes in India who do have their own setups to do mouse models, like the one I'm showing here on the screen, but they don't necessarily cater their services to external clients. It's most in-house based model generation. We do this for external clients. So we have two of these big setups. How this works, we have an inverted microscope here, and you have needle holders. So if you see on the screen here, we have this big holding pipette that's going to hold the embryo. And on the right side, you can't see it here, I'll show you another image. We have a very fine needle that is going to be loaded either with the Crispr reagents, the DNA, the RNA, depending on the type of model that we're generated. And it's going to go and inject directly either in the cytoplasm or in the pronuclear of the mouse embryo. And then we culture these embryos till the next morning to make sure they're survived, and they go to the Twocell, and we do the embryo transfer that I just talked to you about. And then the live pups are born, and we can analyze them. So like I said, there's different types of models of genetic engineering that we can do in mice, and they have different purposes. So it's really important to define your question before you start, so that we can, and that we discuss that with you, to look out at which model will be more adapted to the kind of model that you want for your question. And then once that's decided, then there's different techniques that are adapted to the different types of genome engineering process that is involved in the model that you want to generate. So there's random insertion, transgenics, there's knockouts to invalidate a protein or gene function, there's knock-in to do reporter or to replace a gene in the genome, humanization, so we've done that a little bit to replace the mouse gene with a human gene, for example. And there are still a few people who are doing knockdown, so this is mostly for pre-implantation, but that works well, and we've actually published a few papers with some clients with that. So depending on the type of project that you want to do, and we inject either, like I said, so if I can actually now zoom in, this is the nice part of, of doing this, right? So here you have, again, that one cell embryo, and you see the needle, and we go directly. I'll show you a movie on how that's done. With CRISPR now, we can actually line up some of these embryos in a little microelectrode plate and electroporate the CRISPR reagents in the media. So that blue media will have the CRISPR reagents. And that, we don't need to microinject anymore. So for people who don't have the expertise of microinjection, which is, kind of a high-end expertise, it takes a long time to really masterize. But for simple CRISPR indel knockout projects where you don't need to do large knock-ins, now it's very easy to just do electroporation of the mouse embryos. You can use these little very nice electrodes, which allow you to minimize the amount of media that you use. People do it in cuvettes also. It works well. There's a little bit of more lysis, but the efficiency as well. What you still need to do after that is to do the embryo transfer in the pseudo-pregnant female. The final surgery is still there. So you need to have some people in your animal facility staff who's able to do the embryo transfer. But the genome engineering part, if you have CRISPR agents and you don't have a microinjection technology in your facility, the electroporation is definitely the easy way to go now. And this is what CRISPR has revolutionized a lot. However, for knock-ins and large constructs that you want to do, you still need to do the microinjection. The electroporation will not work. The DNA will not be electroporated to the nuclei of the mouse. As you can see, there's a huge pellucida there. And so we do this either by two different technologies, depending on the insert size. And I'll explain why this two-cell injection. This is a relatively new technology in the CRISPR mouse field that allows us to do knock-ins directly by microinjection. And actually, we've done 16 kb now. Hopefully, it works with this two-cell injection. So basically, we inject both cells at this two-cell stage, and I'll explain why we use this two-cell stage. It's very important. And for still more complicated constructs, we still need the ES-cell technology. So in that case, we have the stem cell facility engineer the mouse ES cells. And once the ES cells have been engineered, we can directly inject the ES cells in the morula of the embryos and do ES cell mediated transgenesis, which is a little bit more time consuming, but for large and complicated projects that is still required. Yeah.
There are a bunch of questions, so would you like to take them now, or
sure, yeah, we can.
Or since they may be also of interest to larger audience, can we? This is a request to the audience that if you could hold on to your questions, we can.
I'm almost done. 2 slides and I'm done. So
sure. I think please continue. Thank you.
So if you have questions on how and do this, we can take this in a few minutes. So this is how. So this is. What I was telling you, this mechanism to do these knock-ins. So it came from this paper from Janet Rosson's lab in 2018. And I've actually interacted a lot with Bin Gu, who designed. And so the idea behind this project is why injecting at the two-cell stage. I don't have the map here. Let's wait. I thought I had that. Yes. So why injecting at the two-cell stage? So in mice, which is very different in humans, I mean, the timing is a little bit delayed. So to be able to do a knock-in, you need to have the homologous recombination happening, right, for your insert to be integrated in your genome. And this usually happens in the G2 phase of the cell cycle. Okay, this is where the chromatin is relaxed and open and open for recombination. If we inject at the one-cell stage in mice, you can see that our G2 phase is very short. What happens in mice is that the true cell stage almost lasts for 24 hours. So we have a big G2 phase here, right? And so this actually gives much more time for the homologous recombination to happen. The drawback is that we have to inject in both blastomeres of the embryo to increase the efficiency, but it actually works very well. And by doing this, we're actually able to knock in much larger fragments than we were when we were doing in the one cell embryo. And so this is a movie. So we actually inject it this way. So this is a movie that I shot when I did a recent microinjection. So you have the 2-cell embryo here, hold it on the left side, and you can see the needle here that's going to go and inject in both of the nuclei of these two blastomeres. And you see the nuclei blow up. That means that the injection is successful and you haven't damaged the embryo. So this is the type of injections we do. It's amazing how these embryos actually take all of this very violent physical stress, but they do well. Actually, if they were going to be damaged due to the physical stress of the microinjection, the lysis happens almost immediately. Within 2 minutes, your embryo just collapse. So usually we wait a few minutes or an hour and we see that the embryos are fine. We segregate the ones that have been damaged or lysed by the injection, and we only take the healthy embryos for embryo transfer. And this is how we have been, so this is another aspect of the 2-cell microinjection that we've increased the efficiency. So instead of using just the regular Cas9 and guide RNA technique, we have this Cas9 that is streptavidin tagged. And we biotinylate the insert. And so what this allows is that now the insert comes as a complex with the guide and the Cas9. So the insert now goes on site to repair as well. Before the template, the guide and the RNA, the Cas9 would be together. That's how they interact. But the knock-in insert is a free-floating element in your solution. So the chances of it finding the target is kind of much more random in that case, and so lower efficiency. With this biotinylated streptavidin scenario, then the insert, everything goes together on site, and so this also increases the efficiency of homologous recombination, and we've had 2 or 3 projects now that work well in this scenario, up to 5 to 6 kb insertions that have been generated this way. This is how we have generated, and we have generated a few mouse models in response to the pandemic. So we've generated some transgenic humanized models. We've generated some ACE2 knockouts as well, and the knock-in humanized using the 2-cell injection technique that I just explained is being finalized. So I think I'll stop here. This is just a table that also this is also one of the good things that come out of the pandemic. So we've been open throughout because we had these COVID projects going on, and we have animals that, don't carry much about lockdowns. They still need to be fed and cage changed, so people need to come. And the staff was amazing about that. They've worked very hard and still are. But what the issue becomes is that we had reagents that had difficulties coming in. So for doing the IVF, the cryo. So we were stuck with some of the media that were no longer being important and accessible. So my MGEF staff were amazing, and they looked at these recipes, which are very delicate medias to do. But we've managed to do all our media in-house, and we quality control them. And now we actually use only our own media, which in some cases turn out to be more efficient than the commercial versions because they're fresh. So that's just there. Training, hopefully we'll be able to welcome you at BLiSC, to see you in person. We've been trying to communicate, to do some of these training via video, and we'll be sharing online courses soon, but it does take time and resources. So we've been a little bit delayed on these online video courses, but hopefully this will come out in a few weeks. And finally, the last slide, but most important slide, this is the staff, the MGF staff. I'm not showing you the animal facility staff. There's 50 people in total here working in the animal facility. We actually are a client of the animal facility. We use a lot of the animals that they maintain for us in proper condition, but the MGF, Cryo, IVF, and Mouse Engineering team is here. We have actually had a lot of turnover this year. Jasper, Debajit, Ardash, and Sonia left because they found some great job opportunities abroad and some other pharma industry here, thanks to the training they received here. And we have the core team here who is carrying over the work. And I think that's it. We have catered a lot of clients here in India, and I'm happy to take the questions that you guys had.
Thank you so much.
Atif, just sorry to interrupt. I suggest we only take a couple of questions now and then move on to Deepti's presentation and then take a lot of questions at the end.
Okay, so quick question. There is one question on the training program. I'll answer it right away. So we try to send out an emailer to our mailing list on the training programs that we conduct from time to time. But the best way is to have a look at the website, and I'll share the link with everyone here. And also there are social media handles of all the 3 institutions plus BLiSC, which regularly posts on the major social media handles like Twitter, Facebook, LinkedIn, Instagram. And we try to keep most of the resources on YouTube now. For the question somebody asked, can we get the slides? We will leave this recording here on the YouTube for you to refer it at a later point. So that's the answer to one question. And then another question, which is from an anonymous attendee, is what exactly is a pseudo-pregnant female monkey?
Yeah, so that's a good question. So the pseudo-pregnant female is actually one of the most important elements that we use here at MGEF. So a pseudo-pregnant female is the female that's going to carry out the pregnancy of the embryos that we have in a dish or that we have micro-injected. So in order for that to work, we need to make sure that the female does not carry the pregnancy of her own embryos, right? So the way this is done is that we mate in mice, we mate the females that are in estrous cycle, and we mate them with vasectomized males. So that the vasectomized males will not contribute to the sperm, but what happens in mice is for the pregnancy to occur, there needs to be physical mating. The hormonal stimulation is not enough. There needs to be physical mating and for the pregnancy for the female to initiate the proper hormone signals. And so for that, we use the vasectomized males. And then the next morning, when there has been physical mating, there's a way to check that the mating has happened because the female vagina would be filled with a little bit of plugged seminal fluid. And then we use these females to transfer the embryos that we want the female to carry out as a pregnancy. So it can be the IVF generated embryos or the transgenic injected embryos. And like that, we're confident that the pups that are born are the embryos that we implanted in the female and not her own wild-type embryos. So that's what a pseudo-pregnant female. It means that she's not pregnant with her own embryos, she's pregnant with the embryos that we have transferred in her.
Okay, hopefully that answers the question. I think there are a couple of longer questions, but I'll jump the queue and ask the one which is maybe a short question. That is, which instrument is used for micro-injection?
For micro-injection, so it's this set. So there's different types. So what you need is a good microscope, but not a fancy confocal microscope. You need a microscope with a very good Norowski or contrast phase image to be able to, you saw the injection thing, you need to be able to see the pronuclei of the embryo, right? So this is all phase contrast, but very, and it's 40x, so we do the micro-injections at 40x. And then you need micro manipulators on either side. And this is the joystick. So it's kind of like a video game, right? You have a left micro manipulator, a right micro manipulator. You have one that's going to be used to hold the embryo with a holding pipette, and one that's going to be used to hold a micro injection pipette in which you're going to go and inject the solution. This here is on an anti-vibration table because obviously you can see if your embryo moves, it's going to be difficult to inject properly in the embryo, in the nuclei. You also need, this is a femtojet. There's different types of injectors, but you need air pressure on your injection site to be able to release at the right concentration, at the right pressure, your DNA into the nuclei. So a microscope, micro manipulators, an air-based injector. Here on the holding side, it's an oil-based manipulator, and a good phase contrast optic to be able to see the pronuclei inside your embryo. Yeah. So there's different versions of this. This is a motorized micromanipulators. Narshige has some manual manipulators. They both work. We have both actually in the facility. And usually people just end up buying the equipment that they've been trained on, and they stay on that. But yeah, both work well.
Thank you, Lily. I hope that answers the question. I think one quick one. I think it may need a bit of prior context from the attendee. Why is it tagged with streptavidin? If you could, remember which one is it?
Yeah. Okay. I went a little bit fast, assuming that you guys knew what CRISPR, how CRISPR works. Maybe Deepti will explain. But for CRISPR to work, you need the Cas9 protein, right? A guide RNA that's complementary to the region that you want to target, right? And so on the left panel is the normal scenario. What happens is the Cas9 protein actually makes a complex with the RNA via this little stem loop here. And then the RNA is going to drag the protein at the right target site and make the cleavage, right? So this is fine if you're doing a knockout or an indel. If you want to do a knock-in, you need these two solutions plus a template of DNA that you're going to insert at the location, right? But this repair template, if you use it in this scenario, is not linked to the protein or the guide RNA. It's free floating in the solution, right? So statistically, for the template to actually find the location it needs to go in on the whole genome is a little bit more complicated. So this technique allows the whole complex to come together. So because streptavidin and biotin are very covalent bonds, they make a really tight bond. So in this context, what Bingu has designed is they still use the guide RNA, the streptavidin, but he has modified the Cas9, but he has modified the Cas9 to have a 5-terminal streptavidin tag. And now our insert, we actually biotinylated by PCR biotinylated primers. And now the insert is going to bind to the streptavidin of the Cas9 via its biotin tag. And now the whole complex, Cas9, guide RNA, and insert that you want to knock in are all going to go on site where they're supposed to go. And this is increasing a lot the efficiency of the knock-in integration. So this is why we use this. It works very well, actually. Yeah.
Okay, I know Dr. Prakash and Mr. Nomesh have asked questions, and Rahul Gupta also, but could you just hold on as we have another speaker with an exciting talk to present, and I will just hear from Dr. Deepti about the most exciting field of Drosophila biology. Classically, this is the first documented model for studying genetics, and for over a century, this has been the most go-to organism, beginning to understand what genetics is, to doing genetic engineering, and of late, the genome engineering is done in Drosophila, and you could actually study the basic physiology, biology, disease biology, and you can also test therapeutic compounds in Drosophila. So with this exciting background on Drosophila, I'm happy to welcome Dr. Deepti Trivedi, who did her master's in at NCBS and PhD from the University of Cambridge, UK, followed by a postdoctoral stint at University of California and Texas. And she joined the campus as the head of Transgenic Fly facility in December 2013. And since then, she has been able to establish and actually move the Drosophila facility to its new avatar of offering genome engineering to a huge variety of clients, both internally and externally. Welcome to you, Deepti. And we are happy to learn from what your facility and your group does here at the campus. Thank you. Over to you, Deepti.
Thank you, Atif. Thank you so much. Just want to know if everybody is able to see this in on the side, genome engineering in Drosophila melanogaster. I mean, is the sides getting cut? Atif, can you confirm?
I can see, yeah.
Okay, so I'll just...
One more confirmation from somebody, please.
Yeah, I can see fine. I can see fine.
Okay, great. Thank you very much. And thank you all for joining us in today's session. I will start. Yeah, so I'll take a couple of slides to say why we are using flies to understand. So basically you can use Drosophila to understand genetic basis of almost everything, because all the major organs and organ systems and tissues are represented in flies, and they are, although the fly itself seems like a very simplistic organism, the genetic system of flies and the organ systems in flies are pretty complex and can be used as a surrogate to understand ourselves better. Having said that, Drosophila genome is actually quite compact. It is complex but compact, and as you can see here, the number of predicted genes is around half that of human, but the number of basis sequence, you see that they are humans have more than 20 times of the sequences, which what it means is that if you were to mutate a human or a fly at the same time, in the sense that you generate 100 mutations in each, either in fly or in human, there is a higher chance that this mutation is going to hit a gene in fly as compared to human. And any more organism which has a higher number of bases or sequences, DNA material, is going to have lesser number of mutations. So which is very good for us, because we don't want every mutation to hit our gene and cause a disease. But if you use that organism, such as flies, for a model system, it's a very good thing that you can actually manipulate the genome even randomly and be sure that it will hit a region which can be of interest. And so historically, that has been exactly what Drosophila biologists for over 100 years have been doing. So what you do is you just mutate the fly, you take a bunch of fly, 100,000 flies, which is easy to do in this organism, and then you mutate it by either X-rays or any chemical mutagen. And what it will do is it will have a large number, all the, let's say, sperms of these flies will have mutations. And so by doing what we call as forward genetic screen, we can find out very quickly what mutation is causing what kind of phenotype. So for example, if you wanted to learn everything about vision, what are the genes involved in visual transduction, you would do such a process where you'll randomly mutate the fly and then see if the fly can see, for example, and then you will have bunch of, let's say, 100 mutations or 100 flies which will not be able to see, and we will find out that these are the genes which are important for this particular phenotype. So this kind of experiments which have been done and have been responsible for several Nobel Prize winning discoveries has been done in flies. The only caveat or problem with this strategy is because it is random and these are random point mutations. It has been very difficult to find the gene which has actually gotten mutated, and it required a lot of genetics to figure that out. So this is something which is still being done, but the only thing is that it's slightly difficult to achieve. So the second thing, which is again still being done, and that's why I want to put it here, is actually insertion of some big DNA element which can be then found or which can be then tracked within the genome. So for example, here you have a transposon, which is a naturally occurring material in different organisms. Here, which we call as P element, if P element can be in presence of transposase, it acts as a jumping gene. When it is active, it goes to different places in the genome, most likely randomly, although it has been shown that it's not that random. But it can basically go anywhere in the genome, and then if you remove the transposase source, it will sit there. Now, if it has inserted itself in a genetic in a region where there is a gene, it is going to interrupt the gene, and hence you can study this. The advantage of P element over random point mutation is, because you know the sequence of this P element, you can always find what is on the either side of this P element and then figure out where the mutation is. So this is something which we also do in the facility, where we have these P elements based transgenesis. Now, the advantage of P element based transgenesis is that we can choose what this P element DNA will contain. So if P element DNA has something which is a reporter. So for example, in this case, you see there's a LAGZ. It can, if it inserts in a region between an enhancer and a target gene of at a locus which is between the enhancer and a target gene, what it is quickly able to achieve is that now this enhancer is going to cause transcription and translation of this LAGZ instead of the target gene. So it's achieving two things here. One is that it is not so the target gene is not being produced, not being transcribed and translated, which means that it is not going to, this is going to be a mutant in this particular gene, but at the same time, using the enhancer and a reporter, you are able to quickly find where this gene is expressed. So there are two examples here where you see that there's a specific pattern in the embryo, in the Drosophila embryo, which a particular gene is expressed. And then the second example is a brain expression of a specific organ, which is important for learning and memory, which is mushroom body. So if you use such a technique where you have basically taken a P element, randomly inserted it everywhere, and then looked at its expression pattern, just because your P element had a reporter, you are able to very quickly, instead of 13,000 genes finding out what is important for learning and memory, if that was the question you were asking, you can very quickly narrow down your approach and find out that, okay, I am interested in all the proteins, all the enhancer traps which express only in these mushroom bodies. And let's say you found 100 such genes, and then you can try to figure out what function these 100 genes have on learning or memory. Now, this will certainly narrow down or make your screen very biased, and you will miss out some proteins, but it will reduce the work. And this has been responsible, this kind of techniques have been responsible for our understanding of many such pathways where the genes are expressed in particular tissue. So not just particular tissue, which is what enhancer trap does, where you can see the expression of a gene, but you can also make these something called protein traps. So in protein traps, what you're doing is, again, the insertion of P element is random, but instead of inserting itself between the enhancer and the gene, what's happening is that the insertion can happen within the gene. Let's say if it inserted in the intron of a particular gene, and if your P element, you have chosen to put not a reporter like LAGZ, you have decided to put a GFP expression, and then the protein that is produced in such a case, if you have added a splicing acceptor, will then express a GFP in tandem with the gene, the protein itself. And then you can find, for example, in this case, if you just see the top panel in this, that some protein is expressed only in the nucleus, which is the first case, or only in the cytoplasm, or only in the in the plasma membrane. So this kind of protein trap experiments are also extremely useful to study. And these are the kind of experiments which can be achieved in flies very easily by random kind of P element insertions. So not just that we, that's one point, but now we are able to also do this in a little bit non-random manner, because so a lot of researchers have generated these lines which can be used by any Drosophila biologist by just ordering them from the fly centers, stock centers, and there are thousands of such lines. So if you were interested in a work of a protein which always go to the nucleus or always stays in the plasma membrane and want to study their function, you can always go to the stock center and choose the flies that you would like to work on just based on their expression pattern, which is by enhancer trap kind of experiments, or their localization kind of experiments. So these two are possible just because the sheer number of resources that are already available to fly biologists to choose from. So you can very narrow down the kind of genes you want to study if you have a very clear vision of what kind of phenomenon or pathway you want to study. So now, having said all that, I would come to what the fly facility is able to do. So, as I said, we do generate transgenic flies, and this is a count from 6 or 7 months ago, but we have generated a large number of these transgenic flies, which are either which function like enhancer traps or protein traps, although we generate them in a slightly different manner, that we actually PCR out the enhancer region and then attach it to a reporter and then introduce into the flies. So this is not random anymore, because this is much more easier to perform. So the way we do that, the way we help the users to generate these transgenic is that we have through CCamp, we have a Drosophila transgenic service, and the user can provide us DNA to generate these, and we generate these transgenics for them. So we can generate transgenics either by random insertion, which is through P element. The disadvantage of having the P element based random insertion is that, again, although you may want some kind of reporter or some kind of enhancer trap, you may realize that this has inserted in some other random gene which may show a phenotype and which may not be caused by your transgene alone, but through its insertion in a region where it shouldn't have inserted, right? So we, okay, before going to what I was going to say, I just thought that I will show how we generate these transgenics. And it's a system that is kind of, so we have these embryos, small embryos. So Drosophila embryos are much larger than mouse embryo. They are visible to the naked eye. And we take these embryos and we align them. So as you can see here, these embryos can be around 0 to 15 minutes old, or even half an hour old. So basically, we are only choosing to take the embryos which have not caused cellularization. And in Drosophila, up to 13 cycles of cell division, there are only nuclei and cellular membranes have not formed. So once we align them, it's just to make our job easy. We use a system. So we have the Narishige micromanipulator, which is more manual, and then we using the same kind of injection setup that Lily was talking about. We inject these embryos, and then these embryos are left to grow into flies and then crossed individually to different balancer stalks, and then we screen for the transgenics. In addition to what I was telling, we also use 5C31 integrase mediated recombination in flies. And what we have is, as you can see in B here, these are the Drosophila chromosomes, and we have some specific ATP sites which are sitting on different regions of these chromosomes. So 2A, 3B, 6E, 20C, X chromosome, and second, third, and fourth, and so on. So these are ATP flies, and these ATP flies, when they are injected with a construct which has an ATB in it, can recombine, and this construct is then integrated into the genome of the fly. So you can this is a much more efficient way of generating transgenics using any of these loci for integration. And the other advantage, just not that it's much more efficient, is also that we know about these loci, that there is it will not interrupt any genetic function or any gene function. And if we were to compare 2 or 3 different transgenics, we can actually reliably put them in the same locus, so that we can actually compare, because one transgene sitting in one chromosome, second transgene sitting in other chromosome, we cannot compare them. So these are the kind of loci that we offer, some of them, not all, for transgenesis. So now coming to CRISPR-based genome engineering. So Drosophila is slightly lower in efficiency as compared to mice for CRISPR. However, we are able to achieve this. So as we know, CRISPR works at higher temperature, at 37 degrees or so, with much more efficiency. But we cannot keep the flies at that high temperature. The highest temperature we can put them is at 29 degrees or so, and that too not to grow them nicely. So then the only thing, the way we do it is just do a lot of them. So some of them will have the mutation that we are looking for. So we offer different kinds of services. So on your left, you can see that we can either inject guide RNA as well as Cas9 as a plasmid into a Drosophila embryo of your own interest, whichever embryo you want. Otherwise, we can actually inject the guide RNA plasmid in a fly embryo that already has a Cas9 transgene. So then you only have to inject guide RNA. Or what we can do is we can generate transgenics, which have your guide RNA of interest, and cross it then with the Cas9, so that the progeny of it will have both the guide RNAs and the Cas9, and then you can finally screen them as you would like. So this way we can just for generating mutants, we can either generate small indels or large deletions or homologous recombination. So in the facility, we offer all these services. We design and generate guide RNA, we design and generate homologous recombination constructs, generate guide RNA transgenics, point mutations, large deletions, and endogenously tagged genes. So before I go there, just to let you know that we have created large number of these mutations, and the largest size of mutation generated is close to 40 kb, but most of the mutations that we have generated are within 10 kb or so. And if somebody asks what is the efficiency of mutations that you have got, so there is not very much consistency in this. I think it depends on various factors, and it can range from 0 to 100% efficiency. So it all depends on what kind of guide RNA, the efficiency of it, which location in the chromosome it is, and so on. So it depends on gene to gene basis. So if we have some time, I just want to say that recently we have generated a large number of mutants, and did also, so we are also providing this as a service, a screening of, once we generate these mutations, we can actually screen them for the tissue of your interest. So for example, this is a screen where the guide RNA CRISPR was only expressed in the eyes. And so you can see the eye phenotype. And in this case, it was expressed only in the wings. So the good thing about this CRISPR-Cas9 tissue specific is that the genes, mutations in which will be lethal, embryonic lethal or lethal before the adult, before the fly become an adult, we can actually do these mutations specifically in tissue by using UA's GAL4 system with Cas9. And here I just show that if when compared to RNAi, many a times, not all the times actually, but most of the time, it shows much stronger phenotype. So this is eye size. So you can see there's a reduction in eye size for DP110 gene when RNAi is used, but you can see that with guide RNA, it is a dramatic reduction. In addition, we have been using this scarless integration of mutations where we can insert any DNA basically of your interest in a genomic locus, but we can also screen it. We can put a screening so for example, here we have used DS-Red for screening. So we can put it, but finally it can be actually by genetics removed very scarlessly. So basically only the region that you want to put will remain. But this additional step allows us for screening just by looking at the eyes of the flies. And then all the flies that don't have this DS-Red will have failed to integrate, while the ones which have this DS-Red will have the integration, and then you can finally remove it. Recently, we have tried this homology independent insertion using CRISPR vector. So the idea behind this is that you don't have to create this homologous recombination because the cloning part of homologous recombination is the most time consuming, and there is a lot of troubleshooting involved. Recently there's a group, Norbert Perimon's group, who have used this, where you can just take a donor plasmid and insert it. If you cut this within the genomic, after insertion into the genome, after injection into the genome by using CRISPR target sites, then there is a good chance. So it's a low efficiency thing, but we are happy to work with low efficiency in flies also. So because we can grow so many flies. So if you cut target genomic region and if you cut this region, there is a chance that it will actually go and integrate. And then you can create knock-ins like this. So the thing is that the knock-ins can be either sense or anti-sense, and you can choose the one which you want in the end. So we have recently been successful to do that, although with a very low efficiency, and we are working on how to make it more efficient. So I'll not take that, but then I'll thank the Fly facility. This is our group have been working very hard during the pandemic. And I am now happy to take questions. Thank you.
Thanks a lot, Deepti. Very nice presentation and detailed. I hope it answered a lot of questions which people had in their minds. And of course, it has thrown up a few more questions, which is the whole purpose of this interaction. So I think there's two questions from Saurabh Datta. How can someone get sure that a P element marker has got inserted under the targeted enhancer?
So P element, as I said, it's very random. It will not integrate itself in the locus that you want. There are 2 or 3 ways to actually achieve this. One is by CRISPR. If you want your whatever GAL4 or LAC-Z or whatever to be inserted in the region which is right after the enhancer or promoter, you can do that by CRISPR. So that will be the targeted approach. And the other is by transgenesis, where you actually take the enhancer region, promoter region by PCR, and then make a transgenic out of it. And then you can put it anywhere you want, basically either by 5C31 integrase mediated transgenesis, or you can put it randomly also. But by P element, you can't, just by P element hopping, you can't put it in a specific locus.
And I think it's a linked question from Saurabh. Any lead on making knockout transgenic in flies? I don't know if you need a further clarification on this question.
Maybe we can, I mean, he can write an email to me about this. Yeah.
Sure,
I mean,
sure.
if he wants to generate something, and we can talk.
Atif, can we put him on
correct. That's what I was suggesting. Saurabh, if you are there, and can you raise your hand so that we can have you unmuted and ask the question? Okay, I don't see Saurabh raising his hand. Okay, I guess I'll answer it here by directing by giving our email IDs so that he can. Oh, yeah, Debashini, yeah. Debashini.
Yes,
you were saying something.
No, I'm fine.
You raised your hand.
I raised my hand. Sorry about that.
Okay. There was a question by somebody on mouse. Lily, you have answered it, I guess.
Atif, there is a question on YouTube.
Sure.
It's asking, what's the efficiency of mutation in a 40 kilobase insert?
This is for which one? Fly or?
I think,
okay,
yeah. So the efficiency was around 20% for F1. And if I remember it correctly, around 4 or 5% in F2s. So there is another linked question. I can see what is the F1 and F2 in efficiency graph. So because this will also help the, so when we do the injections, the progeny, so after the injection, we cross them to a balancer fly. And the progeny of that is an F1. These F1s are not true mutants. They are mosaics. And which means that many of their cells will be mutant, while many of them will not be mutant. So we are hoping that some of their eggs or sperms will have the mutations at this stage. Now, if you do a PCR on these F1 flies, there will be much more flies which will show as positive mutant, simply because there will be cell in each of the fly, hopefully, which will have a mutant band. But once these F1 flies are then crossed to the next generation, only the flies which have, then you will get true heterozygous mutants at that stage. So those are F2s, and they are much lower efficiency, because then also there might be some kind of competition between mutant, non-mutant eggs or sperms or whatever. And then the progeny that you get, they are much lower number of flies in that case which have the mutation. So that's the difference between F1 and F2 efficiency. And and for 40 kb, it was higher in F1, and then by F2 it becomes like 4-5% of the flies with mutation.
Okay, a similar question. Oh, sorry. I think. So from Payal, there is a question on what is F1 and F2 in efficiency graph. It's referring to perhaps your.
Yeah, I just answered that question.
Oh, sure. Thanks. Another next question is how to know this Drosophila model is not lethal for humans?
So Drosophila models can be lethal to humans. We'll not just see them. I mean, we'll not see those humans, developing. So all the developmental genes that that one studies, I mean, those are the genes which even flies will not come out of it. But then every gene will have some point mutation which is not completely lethal and may have an effect on its either development or growth or anything. So we usually will see only humans for which gene was mutation in only those mutations which were not lethal, right? But flies, you can achieve all sorts of mutations just because we can put them either under balancer, so we can keep it in heterozygous situation, or we can actually do a tissue-specific knockout. So you can actually keep the entire body wild type, but only a specific, let's say, wing or whatever you are interested in, eye or whatever, to have the mutation. So you can study the genes for which human counterpart will be lethal, but you can study the function of that in flies.
So I have a sort of a linked question, and that's to Lily. So similar to the mosaic model that we have in Drosophila, where you could have lethal mutations making a mosaic of cells, what's the nearest equivalent in mouse model for the similar thing?
Yeah, you can achieve similar things in mice. Obviously, the numbers are different, but so you can have chimeric. Actually, with CRISPR, you have a lot of that initially if you look into the in vitro, because you will have a lot of mosaicism in CRISPR. Another way to achieve that is with the CryoLock system. So if you use the CryoLock system, you will have a dormant mutation, and then you cross your animal to the Cre, and only the cells that have both the Cre and your flox allele will be positive or negative or knocked out, whatever the way you design it. And the neighboring cells that do not have the Cre, and this is playing on the doses of Cre. So there's actually sometimes having not too efficient Cres is nice, because in the same tissue, you can have clones of cells that have recombined next to clones of cells that have not recombined.
Right.
So you can have tisular control, but it's more like, it's not, it's more by chance that you have these things. You kind of play around with the doses and things like that, where in flies it's actually genetically engineered to be in that state. But you can achieve some mosaicism in mice playing with doses of the reagents. Yeah.
Thank you so much, Lily. There's a Meghashri Nayak has raised her hand. If we can unmute her and she can ask her question.
Meghashri, you can turn on your mic now.
And video also, if possible.
Attendees don't have video, Atif.
Oh, is it? Okay, sure.
You're muted, Meghashri. I don't know if you're speaking, but you need to unmute.
So maybe we can go to someone else until she figures it out.
Yeah, there's a Suresh who has his hand raised as well.
Yeah, Suresh, you can also unmute yourself.
Okay, so I guess yeah, Meghashri, Suresh is going to speak now.
Yeah, I'm asking with Trivedi that can I, if I want to establish a fly facility for testing our drugs, Ayurvedic drugs, can I get a training directly at your place whenever it's a lockdown is open?
Sorry, this was for who? I didn't understand the
It's for
beginning.
Deepti.
Oh, okay.
Sorry.
Oh, I somehow thought that it was for Lily and I didn't hear the question. Could you please repeat? I'm sorry.
Yeah. This is, we are having a non-profit organization, Ayurvedic Research Center in down south in Madurai. So I'm trying to establish a Drosophila facility for testing of Ayurvedic drugs. I need to get some hands-on training directly, if it is possible. What will be your timeline or something like that?
Absolutely. We do these kind of trainings, and we usually do them on one-on-one basis. So we have a form on our website which you can fill, and based on your requirement, whatever you want to learn, you can put exactly your requirement, and then we can plan on how long will it take and so on.
Okay. Okay. Thank you. Thank you.
Yeah.
We have this similar system for our facility if people are asking. So whenever hands-on, whenever people are able to visit our campus, all of these things will be possible. Yeah.
Perfect. Yes.
Thank you.
Thank you both. So I think as a follow-up, we will share these resources from our various websites for you to have a look. And this session will be available on YouTube for people to refer back. There is a question for Lily from YouTube, from Tanya Paul. Can we have a knockout mice for factor 8 in albino mice to make it homophiliac?
I don't know the specifics of the gene, but yeah, I mean, you can knock out more or less anything you want. So the thing that you have to be careful when you make these knockouts is that if the gene is essential for development, then it's best to maybe do a conditional knockout. But that depends on that we can discuss. So it's basically, sometimes CRISPR is so efficient that you will knock out both alleles in the same time, and then if the gene is required for development, you will never have live animals coming out. Sometimes, so this is where the conditionals are important, or to lower the dose of CRISPR. But basically, you can knock out any gene. That's not, there are many consortiums have done that. So you have the ECOM consortium, the COM consortium. They're, obviously in mice to have these full genome knockouts, it takes consortiums and years. But I think the IMPC consortium has knocked out almost, I don't know, 15,000 open reading frames. So that's you can go on their website. We can share the link. But if you have a specific model of a specific region of your genome that you want to delete or inactivate, that's totally feasible. Yeah.
Thank you. Okay. There's another question from YouTube from Ms. Nazia Ansari, and that's for Deepti. Is it like in general the CRISPR is less efficient, or it's a thing in fly alone? What about CRISPR on mammalian cells?
Yeah, so CRISPR actually was first done in mammalian cells, and we have just taken it from mammalian cells to flies. In flies, it is less efficient. I'm sure it is less efficient in other cells also, just because homology helps in putting the DNA in the right place and right orientation, while just having a random piece of DNA floating around in the nucleus, it may go anywhere and bind anywhere. So one needs a much more screening as compared to homologous recombination, of course. But it has been tried in both the systems. And it has been taken in flies from a mammalian system. So, but it is definitely less efficient in flies as compared to mammalian cells.
Thank you. I think we are closing on to 1.5 hour mark, but there are 2 more questions, and I hope both the speakers would be willing to take it. One is, and I don't know whether it's for Lily or Deepti, so I'll read out the question and you may choose to answer. What is the difference between enhancer promoter trap and protein trap, as ultimately both show readout as protein itself?
I can take that.
Sure.
So enhancer or promoter trap will actually give you the expression profile of a protein or a gene. So it will basically, whatever you put as an enhancer trap, if you want, whatever reporter you put as at the enhancer or promoter trap, it can be anything GFP or LAGZ or GAL4. Ultimately, it will form a protein, but then it will, wherever that enhancer is active. So for example, if the enhancer is only active in brain cells, neurons, so then it will, your reporter will be expressed only in brain cells. So that's the function of enhancer or promoter trap. Protein trap, on the other hand, will have this tag, which can be anything, again, GFP or flag tag or any tag. It's actually in frame with the protein, and it can be actually the full protein, or it can be just the N terminal of the protein. Usually people put it in one of the introns or right before the stop codon, so that's their choice. But the idea of protein trap is that within the cell, where is it going? Where the protein is going? Is it going to the membrane? Is it going to the nucleus? Is it going to the endoplasmic reticulum? Just the hope is that whatever region of the protein that you have kept intact will have the signaling region, signaling peptide, which is important for it to be localized in an intracellular context. So that's the two different roles. Actually, it's completely, it serves completely different function, both enhancer or protein trap.
If I can reformulate that also, the enhancer trap is to try to understand how the gene is regulated, where and when the protein will be expressed. The protein trap is actually, you're looking at the protein itself and you're tagging it so you can follow the protein around, right? So one is really to understand how the gene is controlled, and one is to understand where the protein is going.
Thank you both. I guess it's a great answer. One last question from another anonymous attendee. It's about biotin tagged sgRNA and Cas protein strategy for knock-in. Does it work in fly?
I don't know if they've tried in flight, actually.
Yeah, I don't know, and I actually didn't even know about it. I have just marked it that I should read more about this biotin tag because it will be very useful. So thank you, Lily. Yeah. It's 2018. I feel kind of ashamed that I didn't know about it.
So the only caveat with the system is that you have to biotin your insert, right? So we do it by PCR. So there's a limit to the size of the PCR amplification you can do. And the Tristavudin tagged Cas9, we don't have it as a protein because we inject the pure constructs in CRISPR, right? We inject directly the protein Cas9, the guide RNA. So I think you guys mostly do a plasmid mediated or RNA, no? You do RNA, no?
No, we are doing plasmid mostly.
Yeah.
And we are
in this case, it would be a little bit, the plasmid would be fine. Because we do the Cas9 tagged streptavidin as an RNA. We inject the RNA. We don't have the protein yet. So hopefully the protein will be available in that case. It will be even more efficient for us. Yeah.
Good.
I would like to try, so let's see.
You can, we have the RNA here. We can give it to you. On the plasmid, actually. Yeah.
Awesome. I think thanks to the attendee for starting the discussion, and I guess that's the most exciting part about these interactions is that we get newer ideas and new questions. So thank you all. And there's a question or rather suggestion from Shivangini Banerjee for Lily, if you could share the links related to training programs in mouse genetics.
Yeah, it will be on our website and on the BLiSC training program website. For now, there are none because we're locked down. But we are working on doing some kind of hybrid workshops, both for the animal part and for the MGF part, hopefully in July, August. So just keep on watching the links on the BLiSC website or on ACRC website. But these will not be hands-on workshops for externals. They'll be hands-on for local. And online for external.
Thank you. And I think with that, Wish, let me just have a quick look if there is any question on YouTube. No. Okay. So hoping that we will soon be at a time where we can have in-person meetings and trainings. I would like to thank all my colleagues, Deepti, Lily, Debarshini, Chandrakant, and the BLiSC comms team for really putting this together and having so many people join in this morning. We had over 100 people with joined in here on Zoom as well as YouTube, and some exciting questions asked. So again, thank you very much, all of you, and over to Debashini for the concluding remarks.
Thanks, Deepti. Thanks, Lily, for a fantastic session today. And I think the kind of questions that we got, as Atif correctly mentioned, that they really are testament to how exciting the session really was. And thanks, Atif, also for giving us that overview of CCamp right at the beginning. And so we are hoping that we will hear from you will hear from us very soon regarding the next CTIS session. And so with that, I think Chandrakant, also thanks to you for helping us hold these sessions on a fortnightly basis. I think without your help, I think that would have been really, really difficult. So I think thanks everybody. And I think with that, we can close today.
Thank you so much, everyone. Have a great day.
Bye. See you.
Bye.
Thank you.